and welcome. Thank you for joining us today. I'm Sara Abtahi, a dentist and PhD researcher in dental biomaterials. Today, we are having Dr. Rajdeep Chakraborty presenting the very interesting topic of cell culture techniques for application in the oral and maxillofacial region. As you know, the regenerative strategies continue to advance in oral and craniofacial healthcare, and three-dimensional cell culture models are gaining attention for their ability to be better mimicking physiological environments compared to two-dimensional methods. Dr. Rajdeep Chakraborty is ACCRF-NIH Fellow and also academic at School of Science and Engineering at Macquarie University. Before we begin, just a few quick notes. This session is going to be recorded and might be shared, so be aware of that. Please keep your microphones muted, and you're welcome to submit your questions anytime in the chat bot. And if you don't mind, I would like to have some quick notes in Farsi. میخواستم که مجدد سلام و عرض ادب داشته باشم خدمت عزیزانی که شرکت کردن. به عنوان عضوی از خانواده دانشگاه علوم پزشکی تهران برای من باعث افتخاره که مدیریت و مدیریتور بودن این جلسه رو بر عهده داشته باشم و خواستم تشکر کنم از همه کسایی که تو این شرایط دارن امید رو تو دانشگاه زنده نگه می‌دارن. And without any further delays, we are all ears now.
Okay. Salam to everyone, and this is officially I'm known as Dr. Rajdeep Chakraborty. Otherwise, my unofficial name is Wasim Wali Ahmed Khan, and I'm from India. And currently, for the last 7 years, I'm at Macquarie University, Sydney. And let me give a brief introduction regarding my background. So I've done my Bachelor of Dental Surgery and completed in 2012. I completed my 4th year in 2012, and then I immediately, because of my marks, I got Indo-German Academic Exchange Scholarship to do my internship for the 5th year in Germany, and also in Calcutta Medical College and Ahmed jointly. And that was the foundation for my dental alveolar surgery, part 1 maxillofacial surgery. And then I had a small tryst of clinical experience and as a house staff in All India Institute of Medical Sciences, Calcutta Medical College, and other places. And then I did my MSc from Lancaster University, UK in 2017. I completed that, a 1-year course. And immediately because of that, I got my MQ Research Excellence Scholarship to do my Master of Research. And I applied for a grant to have my own oral cancer research here. And from Sydney Vital Research Scholar Grant. And I completed my Master of Research in 2020. And again, I got a scholarship to do MQ Research Excellence Scholarship to do PhD in Macquarie University. And at the same time, I got Australian New Zealand Head and Neck Cancer Trudy Shine Adenoid Cystic Carcinoma grant in 2021. And as the first PhD candidate to get an external research grant from NZHNSCS in 20 years. And I completed my PhD thanks to them for funding my PhD. PhD research I completed in biomedical engineering in 2024. And I appeared on the first attempt of FRCS, Fellow of Royal College of Surgeons, and I completed that, the part 1 last year on the first attempt from Edinburgh, and currently affiliated with them. And also last year, and by the end of last year, I got from a pilot grant from Adenoid Cystic Carcinoma Research Foundation, and it has component of National Institute of Health, Maryland. And currently, I'm a principal investigator and a research fellow at the School of Natural Sciences. And I don't know whether I am visible enough or not, but this is an old office, and this is my office from where I'm conducting this presentation. And all this has happened because of me doing 3-dimensional cell culture. Now, what is 3D cell culture? Basically, when we do cell culturing in our lab, then we place cells on plastic. Can you all see my cursor moving? Can you all see my cursor moving? I believe. Can you all see my cursor moving?
Cannot see the cursor moving. No, no, Dr. Chakraborty, we cannot see that.
No worries. I'll just then continue
Yeah.
without moving the cursor. So as on the left-hand side, you can see the 2D cell culture, which we all commonly do, is just by placing the cells on a plastic well plate and then covering it up with media. Media is having nutrients, and that helps to grow the cells. Now, whenever we test any drugs or materials, whatever, we directly deposit on top of it and see whether the cells are dying or cells are growing, cells are healthy or not. But is that enough? It's not enough to see whether the cells are actually, whether it's a real world scenario. It's not a real world scenario when we are human being, and the cells are obviously interacting with other cells. The cells are interacting with immune cells. The bone cells are interacting with nervous cells. The blood cells are interacting with muscle cells. So everything is getting interacted with each other. So the real world scenario is to have a three-dimensional culturing where each cell is interacting with another cell, and the cells are not just on a plastic plate, but they are in the extracellular matrix. So below, as you can see, a conventional 96 well plate uh, where the 2D cell culture is done. But here, the 24 well plate, as you can see, where the 3D cell culturing is done in an extracellular matrix. Now, going into the what are the components required for 3D cell culturing? For 3D cell culturing, basically we require four things. First, cells, of course, but the cells can be primary or cell lines. What's the difference? The difference is the primary cells are those cells that are directly taken from a subject, from patients or from healthy subjects. But cell lines are immortalized. Basically, when TERT is overexpressed, they don't tend to die. But the primary cells that are taken, they die. We can't maintain their growth. So they can be cultured in a three-dimensional environment. And what is required for that? Extracellular matrix. We can buy, or we can collect it, or we can make it our own. And that's what the revolution of material science is, where it is amalgamating with chemistry and engineering, and that's how we can synthesize our own extracellular matrix. We can collect it definitely from the patients, from directly from the patient. We can buy it, we can purchase it from companies. What are the platforms? Now, as we have seen in the previous slide, that there are 24 well plates, there are 96 well plates, they're all plastic plates on which we can drop extracellular matrix, and then on top of it, we can place the cells. But is it enough? Is it the real thing? There, where again the material science coming, where we can make our own platforms, and those platforms are nothing else but scaffolds. We'll come to it later. And what else is required? We require food. Why? Because each of the cells in our body need food, energy, and that is from the media. So we all buy media, but can't we make media? Is the media enough for actually mimicking the real world scenario? No, the answer is a big no. It needs a lot of additives, a lot of supplements, and that what reverse engineering or proteomics, the power of proteomics is. We have to, or we ought to understand what exactly the components are there that are interacting with those particular cells. We'll come to it later. So, what are the techniques of three-dimensional cell culture? There are basically two types of techniques, a non-scaffold based and a scaffold based. Let's go to the scaffold based. Now, having extracellular matrix, that itself works as a scaffold. If it stiffens, it can take any shape. It can take the shape of those bottom surface of the well plates. It can take a shape of any molds. If we have a mold made up of PDMS or anything, it can take the shape of that. Then we have cell culture inserts. The inserts directly can act as three-dimensional environment. What are they? We'll come to it later. Scaffolds, something that even Dr. Abtahi is doing, something that even we all are interested in, making scaffolds directly. And then what we do is spheroid formation. When we take all these primary cells or even the cell lines, we place it in specialized spheroid plates. We drop the cell culturing over that. They tend to conglomerate, they tend to aggregate and form spheroids that we can see here. They are actually three-dimensional cell culturing, one of the first steps of three-dimensional cell culturing. And then there are organoids, as we can see here. The organoids are basically, we get it from the stem cells. They are pluripotent in nature, and they can transform into any sort of shape and structure, and even metabolic properties changes. Then comes microfluidic devices, and that has revolutionized the three-dimensional cell culture. Why? Because blood is flowing everywhere. So the media should not be stagnant, should not be static. It should be moving. But how can we make media moving? We can do that using, as you can see here, the plates. They are actually designed plates that are programmed, and that can generate the real world scenario where microfluidic pump is attached to this kind of plates, and there is peristaltic movement of the fluids. So that is very, very close to the actual real world scenario. Bioprinting. The bioprinting is three-dimensional bioprinting. When we feed the computer or the system with a particular design of the scaffold, and the nozzle, the fluid or the extracellular matrix with the cells come out of it, taking the shape of that particular scaffold or forming a particular scaffold. That's what 3D bioprinting is doing at the moment. Now, what are scaffolds? Scaffolds are basically the organ-specific histoanatomical elements like vasculature circuits and parenchymal and stromal compartments. Compartmentalizations are preserved. So scaffolds are basically nothing but the supporting structure. They are the mechanical supporting structure. That means anything that is preserving the structure, devoid of the cells. For example, bone is having a scaffold, as you can see here. Liver is having a scaffold, as this has been developed by even us, even one of my collaborators here at Macquarie University, where we are taking a piece of the liver, and with our technique, we are actually getting rid of the cells, but we are keeping the structure. If we get the rid of the cells, but keeping the structure, then if we add our cells according to our choice, it can be grown. It can be done in a lab. So scaffolds help doing three-dimensional cell culturing. So how to make a scaffold? Yes, we can do it. There are 4 major types of making a scaffold. 1, directional freeze casting. Second, immersion agitation decellularization. And that's what I'm going to say now. Third, we can do directly 3D bioprinting to create scaffolds by feeding the computer with G coding or STL designing. We can actually direct the machine to drop or to make the fluid flow to a particular, taking a particular shape, and supporting structure based. And that is, if we have if we can increase the stiffness of a particular extracellular matrix that we can synthesize ourselves, or we can even purchase, then we can actually create, with the help of a mold, a scaffold. So let's create tissue engineering constructs. Here, what we have done recently, and this has been done by me recently in Macquarie University in collaboration with Dr. Anna Guller. We collected sheep's tongue, And we have collected it, and we have taken the samples from Macquarie University, and we have used decellularized and sterilized the particular sheep tongue. And because there are two types of scaffolds, mucosal and stromal. Here we are reseeding after two-dimension cell culturing, because we need to culture the cells first and grow the cells in two-dimension cell culture. And then taking the cells from there, we are reseeding or replacing the cells in those small pieces of decellularized sheep's tongue. Here we used SCC4 and Cal27. They are oral squamous cell carcinoma cell lines into all these mucosal and stromal scaffolds. And this is the pictures that were taken by Anna while she was doing it, and I was just beside him. The ships, as you can see, we have used biopsy punch. It's very easily available in the market, and we have taken the ship's tongue from butcher shop, and we have actually taken the punches of those ship's tongue, and we have decellularized it, and finally we have basically using our sterilization techniques and the agitation procedure using different detergents, we have cleaned it up and made it ready to get seeded with the three-dimensional cell culture cell lines or primary cells. Basically, in this case, we use the primary cells later on, so it is working, but in the initial stages, we used immortalized cell lines. So here you can see how we have sterilized the scaffolds and overnight conditioning with fetal bovine serum. That is a very basic nutrients used for culturing cells, and containing media, and sterility check was done. And after 2 hours of addition, after the seeding of the cells, because it nearly took 1.5 to 2 hours in the incubator, 37 degrees Celsius and 5% carbon dioxide. So we just added 1 mils of the media. So very simple, and it can be done so anywhere. So what are cell inserts? Why am I talking about cell inserts? Because they are all through till now I've talked about direct cell culturing, but there is something called indirect cell culturing. When you have primary cells from one specimen, and you have cell lines. Suppose you have immune cells that are cell lines, so they are HLA optimized, so they are not going to react with another cell line. But when you have primary cells from two different sources, or when you have primary cell line from one source and cell lines from another source, then you have to do indirect cell culturing, and there where we are using this cell inserts. The cell inserts are having a membrane, as I don't know whether you all can see this on the right hand side. This is an insert, and the membranes on the bottom surface, they are interacting with another cell type on the other side, placed on the left hand side, as you can see, placed in well plates. So these membranes, these semi-permeable membranes in the inserts can be made by polycarbonate membrane, can be polyethylene terephthalate membrane. So they are actually facilitating the permeabilization of media, that exchange of media from one compartment to another. And also, depending on the pore sizes of the semi-permeable membrane, it can be from 0.4 to 8 microns. They are decided based on what type of assays we are going to do, whether we are going to see whether the immune cells are moving from one side to another side, or cancer cells are moving from one side to another side. So this is an example model that's been prepared by me and has been successfully submitted for provisional patent last year. So I used an indirect cell culturing, and in addition to direct cell culturing. So as you can see on the bottom surface is a semi-permeable insert, and natural killer cells are the immune cells. And here the media for growing natural killer cells were myelocult and interleukin-2 and host serum. And these additives were for optimized salivary gland media to culture adenoid cystic carcinoma cell lines. So as you can see, using all, and here the collagen matrigel acted as the scaffold, acted as a extracellular matrix for this whole three-dimensional cell culturing. So how do I validate the data? All the data are original and published, and some of them are unpublished. So first, let me give a brief overview of what I did while I used the three-dimensional cell culturing here. I analyzed using microscopic images and viability test and apoptosis assay, whether my technique is right for the cells or not, whether the cells are healthy or not, whether they are growing or not, whether they are at all alive or not, they're surviving my components or not. So I did all these things, and after validating that my cells are healthy, they are happy, and they are not dying, so I did other biochemical assays to acquire data, and I did proteomics assay to see what protein differences are happening during my work, because I overexpressed and silenced a particular gene to see the function. So I used proteomics. So a model can be used for so many things, as you can see. So here are the example pictures. As you can see on the left-hand side, this UMHSCC2A are the cell lines from University of Michigan. And they have been done three-dimensional cell culturing. And this is the morphology, where the green is the actin cytoskeleton, or the outer surface of the cell, and the blue are the nuclei. And all of a sudden, the aberrant shape, that what got published in the scientific reports, that all of a sudden the shape changed while it interacted with the immune cells, even indirectly. And but the rest of the cells, A253 are mucoepidermoid cell line, one of the another type of salivary gland pathosis, and SCC9 are oral squamous cell carcinoma cell line. So you can see no changes are happening. Even SCC4, Cal27, and SCC25, they are all oral squamous cell carcinoma cell lines, and you can see no changes are happening. But the interesting thing is, as you can see, that from a three-dimensional cell culture model, that's what I actually intend to show all of you, that so many types of assays can be done, and high profile, high resolution, three-dimensional images can be acquired, and that are very, very much presentable and publishable. So OKF6 are normal oral mucosal cell lines. So even I have seen how after interacting with immune cells, if there are any changes. Obviously there will be no changes, because we are normal human being. If immune cells are going to cause any problem, then there will be autoimmune disorder. So this is an example picture of the trypan blue assay. It's a picture taken from the monitor, that as you can see, the live cells are 100% in both the cases, even after interacting with immune cells and without interacting with immune cells, in a three-dimensional culturing. As you can see, in both the scenario, the live cells were 100%. So no doubt that the cells were not dying, or some aberrant changes were not happening because of that, until and unless you make some gene changes or protein changes or add any drug. So I did immune regulation cell assay. They are example pictures of my own results. They are I've done flow cytometry. So as you can see, FOXP3 is one of the T regulatory cell marker, whether the immune cells are getting confused or not while attacking the cancer cells. So, and interleukin-2 receptor alpha and CD25 plus FOXP3 are known as T regulatory cells, or when the cells are stop attacking cancer cells or any sort of virus or bacteria. When your defense cells or immune cells stop attacking, that's a very bad thing. And these are the proteins that get highly expressed that time. So it is all these kind of assays using different cell lines, different immune cells, different cancer cells, and as I would like to play this video. I don't know if I can play this. So this is a 3-dimensional Z-stack immune cells showing attacking the cancer cells. But unfortunately, I can't play this video. Anyways.
I think we cannot play any animation in here. Yeah,
Oh,
sorry for
all
that.
right. It's absolutely
Yeah.
fine. It's absolutely fine. So, anyways, these are from taken. These green protrusions are the cytoskeleton that are trying to attack the other side of the cancer cells. So it has been taken in a very high resolution confocal microscope, and the blue are the DAPI stained nuclei, signifying that they are really the cells that are immune cells that are trying to attack the cancer cells on the other hand. And they are the representative pictures of SWOT, or data independent analysis, or basically proteomics. That's what I did to find out what are the changes that are taking place in the immune cells while they are trying to attack the cancer cells when I actually overexpressed and silenced a particular protein of my interest. And while I got a list of immune cells out of it, immune proteins out of it, I did string analysis to see how the proteins or my interest of immune proteins are interacting with each other. As you can see, so basically my intention to show you all these things are to let you know that how much the 3-dimensional cell culturing can help we acquire abundant amount of data, and that can be translatable directly to clinics. And same here, without antibodies, the proteomics can be done to find out the actual protein expressions using multiple reaction monitoring. And this is an analytical chemistry part that I did, and that really helped me to complete the project, and they are all published. How much is the cost and time to make 3D model? That's a very important thing for all the supervisors and the students, and how much is the cost? So if you purchase extracellular matrix and the other components, then it is $64 that I required to make my thing. But if you try to synthesize or make your own scaffolds, then the cost will go down and down and down. And once you can make a very optimized a scaffold that can actually harbor different cell lines, then the cost effectiveness will be higher. And here is you can all see this later on, how I did the unit economics of the total investment that has been done to make a 3-dimension model. And everything is mentioned here. How much is the instrument cost? How much is the cost of the cell insert? How much is the shipping cost? Microscopy facility? How much do you require to check the viability of the shape and structure or the morphology of the cells? And even how much do you require to grade the cells, make the media, have the growth factors, preserve the cells? Everything is mentioned here. So you can all see this later. And without my mentors and collaborators, it would have never been possible. And I'm greatly thankful to my long-term mentor and my teacher. He's a now he's a good friend of mine, Jax. Jax is the dean of the School of Dentistry of University of Michigan. And he's also a professor of dentistry and also a professor of otolaryngology and head and neck surgery. And my long-term teacher and collaborator, now my official collaborator, both Jax and Mark Lingen. He's a professor of pathology and professor of medicine from the University of Chicago. And he's the head of the Committee of Cancer Biology across United States of America. And finally, you all must have been reading now Robbins' pathology, or must have read it previously. He is my oldest teacher. He's my teacher for the last 15 years and my biggest mentor and supporter. His name is Distinguished Professor Vinay Kumar, and is Lowell, I should be reading this, Lowell T. Coggeshall Distinguished Service Professor of Pathology and directly appointed by the President of the United States of America. He is the editor-in-chief of the Robbins Pathology. And and I'm grateful to Allah that I have been given this opportunity, that I could interact with them. They are my references. And yeah, love Iran, support Iran. Thank you to everybody for listening. Any questions?
Thank you so much again, Dr. Chakraborty, for your insightful presentation. We now have time for questions and answers. Feel free to post them in the chat bot, or you can just simply raise your hand in the raise hand feature. Dr. Dabai, could you please open your microphone?
Yeah, thank you. Hello, everyone. Dr. Raj, thank you very much for your amazing presentation. I have a question. Maybe you have already mentioned it, but maybe I didn't get it. How do you choose the best type of cell to co-culture with the main cells? You know, in your project, I can understand that you're working on cancer cells, and somehow it is obvious that cancer cells have the most interaction with immune cells. But in other projects, is there any special way to choose the right cell to co-culture with the main cells?
Can you please repeat the last part of your question? Can immune cells can be co-cultured with?
Yeah, I'm talking about the best cell type of cells I'm talking about the way to choose the best type of cell to co-culture with the main cells. In your
Okay.
project, because you were working on cancer cells, it is obvious that cancer cells have the most interaction with immune cells, maybe somehow. But in other projects, I'm asking, is there
Yeah.
any special
Yeah.
way to choose the
Yeah.
cells?
Yes. Yes. Excellent question. I would like to say that yes, for choosing the cells and the cell types, uh, what you ought to do is the most important thing is, first of all, suppose you have a project on implant. Let's now move from cancer to implants. Suppose you have a project on implants. So you are trying to see whether the osseo integration is happening or not. Suppose, are you listening, Dr.
Yeah,
Saud?
yeah,
Yeah. So suppose you have that. So in that, the osseo integration ought to be taken with the help of osteoclasts. So the first thing is that the osteoclasts, they are immune cells, your immune cells. Correct. Correct me if I'm right or wrong. So in that scenario,
Yeah,
you would say,
we have
yes,
many cells that we
yes.
may have
So for,
the main cells have interaction with that. Maybe a
yes,
fibroblast, maybe osteoblast.
osteoblasts, exactly. So there, when while you are choosing, so your crux of your question is that when to choose primary cells, what type of cells, and how to understand that how we are going to culture them, direct or indirect. The answer of the question is first identifying what project we are doing, that then identifying what is the anatomical area that we need to see. If we need to see a jawbone, whether the bone is growing or not, so we are going to choose our cell types accordingly. First. Second is, if we are getting the cells directly from the patients. Suppose you are getting directly from the patient some bone cells, and other hand you are not getting the immune cells. Suppose from that same patient, if you are not getting immune cells from the same patient, immune cells, what I mean by this is, you have taken blood samples from the same patient, and then you extract using ultracentrifugation, you extract the that is PBMCs. Okay. Now mononuclear cells basically. So while you're doing this, you are interacting that you can interact directly, direct co-culturing you can do. Uh, with the same patient bone cells collected from the same patients. But suppose you are purchasing bone cells from a cell line bank, and then you are having a patient's from a patient PBMCs or immune cells. You cannot directly co-culture it. You have to do, first of all, you cannot do any sort of co-culturing with the patient derived PBMCs. Patient derived primary cells can be co-cultured with uh, immune cells purchased from the companies, but you cannot do the direct interaction. The reason is, on the surface of the cells, we are always having HLA, histocompatibility complex, or like antigens. What they do, they recognize the MHC complex and they try to attack. So what happens is that when you, for example, the reason behind one person's graft cannot be implanted into another person, the reason is because of this allergic reaction, because of the mismatch. The same thing is going to happen here. Because of the mismatch, a cell from another source, a cell from another source, you cannot mix both the things. So if you are having cell lines only, then you can direct co-culture it. If you are having two different sources of cells, then it is better to do indirect co-culturing using inserts or something. Choosing the perfect cell type, it depends on the anatomical area from where you are choosing it.
Thank you very much. Thank you very much.
Sorry. All this time
Yeah.
I thought I'm speaking. Yeah. Okay, thank you for insightful explanation. And I have one question myself, and I know it's a bit general. What are current limitations of three-dimensional cell culture when you want to relate it to clinical practice?
The One second. Yeah,
What
you
are
can
the
hear.
limitations for
The limitations for the
translating
limitations
into
for
preclinical or clinical? Yeah.
the biggest limitation that everybody is trying to overcome is our blood is continuously flowing. So this, what are the any circulatory fluid, for example, whether we are having periodontal fluid or whether we are having blood in our veins or artery, whatever capillary fluid, everything is mobile, everything is moving continuously. So while we are doing three-dimensional cell culturing, the things are three-dimensional but still very static. That is the first thing. So what microfluidic pump system, the prerogative of using microfluidic pump system, or the benefit is, it is continuously helping or facilitating the movement of the immune cells or other interacting cells. First, that is the biggest challenge. The second thing is that we cannot do pharmacokinetic or pharmacodynamic dynamic analysis using just three-dimensional cell culturing. We have to use a little bit of, at least three-dimensional cell culturing is going to reduce the number of animal usage, but animal usage is compulsory. The reason is to understand the pharmacokinetic and the pharmacodynamic behavior of the pharmacological agents or whatever we are trying to overexpress. And the third and the most important thing is, a lot more development needs to be done, particularly designing scaffold, particularly having a common scaffold, particularly using scaffolds for various purposes and optimizing it. So lot more work needs to be done.
Thank you for your explanation. Is there any more questions? If there is no more, I want to thank you again, and I'm very grateful for you to be here today, and even more so for being here on Saturday, that I'm sure that it was your off day. That's really valuable for us. Thank you again.
Khuda Hafiz to everybody, and I hope everybody will be safe and fine.
Thank you, and I thank every attendees for being here today. Thank you. Bye.
Thank you, doctor. Thank you. I just wanted to thank you and say that it was a very useful and interesting webinar for me. I learned a lot. Thank you very much.
Thank you. Thank you so much. Thank you.